I'm sorry. Okay, so it's I wasn't planning on giving the talk in a Zoom talk, and we are in the middle of a crisis where we actually, my group is working hard while I'm sitting at home and running Zoom meetings. But I'll tell you about something that we've been doing the last 2.5 years, and it's something that's speaking a lot of was speaking a lot of momentum before the crisis, and we hope to continue this. And before I forget, there are many people involved, but 3 of them that are listed here, Ronen, Isra, and Gabriel, are the core team that is pushing this throughout the life of the project. So they deserve, they're all in my group and deserve a lot of credit for what I'm going to talk about. I assume very little knowledge about biology, but if you feel that I underestimate the knowledge of biology, please point to you did, and I'll try to clarify terms I use if I miss something. So I think all of you had probably went to take a blood test at some point in your life. And there are many things we can test from blood. We can look at cell counts to see the immune system. We can look at all kinds of metabolites and proteins. And in the last 3 years, people have started to talk more and more about using DNA as a substance to measure in blood as a diagnostic marker. So why DNA? And so let's return to the basic of biology. The way cells work, we have DNA, which is a sequence. It's like a book. It's a sequence of letters that doesn't do much, but there are people who can read them, or machines that can read them. And the way they're being read is the DNA is transcribed to RNA, and the RNA later on is translated to protein, and proteins are usually the things that are the machines that do work in the genome. But their sequence is determined by the sequence of letters in the DNA. So there are about 20,000, depending on how you count, protein coding genes in the human genome, and many of them are active in all cells because they do what we call housekeeping or necessary functions. But many of them are cell-specific. For example, they are working only in neurons or in the liver, or only when we're suffering from heat exhaustion or something like that. So there are many questions that we can answer about the body by looking at which genes are being used, and many of us are doing it through looking at RNA. But today I'll talk about something that is connected to that, but is not directly RNA. Okay, so why DNA in blood? Well, it turns out that cells that die inside our body there is some recycling going on, and there are multiple pathways for recycling. One of them is release of the debris from the cell into the bloodstream, where it's later filtered by the kidney and the liver. And the number of cells that die each day is staggering. Most of them are white blood cells, but there are many other cells that die. And if we look at the blood of any of us, healthy human being, we will see something like 2,000 cells worth of DNA in every cc. So that's an ongoing process that keeps happening all the time. And when there are extreme conditions, stress or disease or other things, then this amount can increase quite a bit because there is more turnover due to those conditions. So why is this useful for diagnosis? The main way of thinking about it is if there is DNA, we can read the sequence. If we can read the sequence, we can tell something about the DNA. And one situation where it's very acutely useful is when we look at cancers. So one of the hallmarks of cancers is the fact that the cancer loses all the quality control that are involved in replicating itself. It introduces a lot of mutation into the genome. And many of those mutations are important ones because they cause usually a disruption in a protein that is regulating against against growth, uncontrolled growth. So the cancer has to bypass some of the guardians of against proliferation, and genomic change is the most common way of doing that. And many times the genomic change is through mutation, a change in the letter, or deletion of some of the letters, and that's something we can read from the DNA sequence. Because of that, in the last few years, there's an ongoing interest in using what is called liquid biopsy, where cancers is one of the, a growing tumor releases a lot of DNA to the bloodstream. There are companies that already sell kits that extract the DNA, find mutation, and relate them to the status of the tumor. And that's something that is already in practice and will keep growing more and more. However, this concept suffers from a problem. When we are looking at the DNA sequence, in case of mutation, we can detect a mutation because there is a change in the letter. But in all other pathologies, the cells that die are our own cells. They have the same DNA sequence as all of our other cells. And so for all practical purposes, the sequence is the same regardless whether it's a liver cell is dying or a colon cell or just a white blood cell, which is okay, that it's usually turning over or dying at some amount. So for visualization, we can think of it, all the sequences look the same color. We cannot tell where they came from. And so that's why the focus on cancer is more or less the biggest application, because there we can find mutation. So our contribution starts from an old observation that was somewhat overlooked until recently, is that most of those pieces of DNA are not circulating as the images I showed you before as kind of little bands of double-stranded DNA, but rather in a complex with a set of proteins. Those proteins with the DNA, the complex is often called a nucleosome, and it's part of the packaging of DNA in cells. So let's return to one of the molecular biology classic images. If you remember chromosomes from high schools or from various nature or science TV series, are those things that we have multiple, we have 23 pairs of chromosomes, but the chromosome is essentially a very dense structure that's composed of this wire that is built from multiple layers of organization. The basic unit of that organization is those disks with DNA around them, which slightly more realistic, but also fairly not that realistic visualization is this one. So there's a piece of DNA, about 150 base pairs, so 150 letters, wrapped around this core of protein. And this is kind of the repeating unit for packaging the DNA inside our cells. And so when the cell dies, essentially this structure is disrupted, but most of the basic units are kept intact, and that's what protects the DNA in the blood. So why is this interesting? Well, it's interesting because in the last 20 years, many labs, including my own, worked on research on a whole set of modifications that are often called epigenetic modification. They are above the genome, where the cells change in different ways, and I'll skip the chemistry of that. The proteins that are the scaffold for the nucleosome. So these proteins in this, again, illustration as a disk, can be modified to contain different groups of different colors or different chemical entities. And in the illustration below, we use the flags as kind of, so red and green flag, but there are many colors of flags. And the benefit of that is that while the protein is the same, the basic protein is the same throughout the genome, once you paint it with different flavors of flags or flag it with different flags, you can say the DNA that is packed here is not useful for me right now. Let's put it to the side. While this DNA is very useful, we need it in the center and close to the machines that will work with it once the signal is given. And it turns out that this kind of information is very conserved throughout life. It's the same in all the cells up to some extreme special cases. And so the sign that say, for example, that a gene is open for business in a liver cell and closed for business in all other cells is the same in all cells. It just marks different territories depending on what the cell needs. So just to return to this example, there's a gene that encodes for a protein called albumin, which you might heard about. It's produced by the liver. And only in liver cell, it's marked as open for activity. While in all other cells, this gene is not used and it's packed away in a piece of the chromatin that will be in the Boyden, locked in the storage and not used by the cell. And so while the DNA sequence is the same, if we look at the epigenetic marks, they will be different between a liver cell and any other cell, say a neuron or a brain cell here. So this is a change that is more related to the activity of the gene in that cell and not about what the gene is encoding. It's how it's being used. And so our idea is that we, instead of looking at the DNA sequence, we're going to look at the DNA sequence and the marks that are essentially it's packed upon. So we will be able to distinguish whether this DNA sequence comes from an active region in the cell it was in or an inactive region. And so if we return to this illustration of possible ways of looking at blood as a tool for diagnosis, we're adding a whole new media for diagnosis, which is the complex of DNA proteins. We're using the proteins to distinguish what is the status of the DNA, and we're using sequencing to see what piece of DNA is with that status. So before I go into details, and let's think how we are going to use it and give a kind of a sense of the flow. So assume we have several cells drawn very schematically. And only in liver cell, the albumin gene, which is the albumin encoding gene is called ALB, is packed with those green flags. In all other cells, it's packed with those red flags. Now suppose two cells, one a liver cell and one another cell, die. When they do, they go through the process of coordinated cell death, which is called apoptosis. The cell is chopped up to pieces. The pieces of the chromatin are now separated from each other, and they are essentially collected in the bloodstream where they join many other pieces of DNA from all the other cells that died in the recent few months. When we take a blood sample, hopefully we capture all of them in the same way they were in the blood, if we are doing the sampling correctly. And so at this stage, we have a whole collection of nucleosomes with DNA on them, with different marks, but we want to look only at the active ones. And the way we do it is we use a technique that essentially captures only the pieces that have green flags. It's called immunoprecipitation. It uses antibodies against the green flag for those who know a bit of biology. And it allows us to enrich to take a very rich pool and to enrich only to the subset that contains the target, in this case, the green flag. So after this enrichment, we are left only with a subset of the nucleosome, but it's a very special subset because we know something about it. Now we can take those enriched nucleosomes, go through the process of sequencing DNA, which is today something that is almost standard to do sequencing of many small fragments of DNA. And then we can take the pieces of DNA and search where they are. This schematic is the genome, where they match the genome. and we can kind of pile them up along the genome. And usually we will just show not exactly the fragments, but just how many fragments are covering each part of the genome. So this will translate to a somewhat funny looking peak here. We're going to call this peak. And this is essentially saying there are many reads that were aligned to this piece of the genome. Now, if this piece of the genome happens to be the albumin promoter, then we can put 1 and 1 and say these pieces of DNA came from active genes. The gene is active only in liver cells, and therefore we have evidence that the subject from whom we took the blood suffers from death of liver cells. So I hope this is the main idea. So if there are questions, now is the time to ask if something was not clear about the concept before we go into the details and how it works. Any questions? So I see one hand. You're handling this?
Yeah, I just allowed this person to speak. But are you interested in asking a question?
Okay, so in the
we have a
meantime,
question from Yuval Abani. He asks, why do you need to sequence the DNA from the fragments?
That's a wonderful question. If we don't sequence the DNA from the fragment, we can do things like count how much DNA we found, but we don't know where it was from the genome. When we sequence it, we essentially get a sequence of letters, and usually there is only one place in the genome where we can find those letters within a piece of 150 base pairs or 200 base pairs. That's supposed to be the length of the things we're sequencing. So the sequencing gives each fragment of DNA essentially an identity, where it came from the genome, and this we can tie to what gene was there, and therefore we can make the connection to function. We are not sequencing the entire genome. We're only sequencing. The question was, why sequence the whole genome? We're sequencing only the enriched fragments. It's a small fraction of the genome. That's actually one of the benefits of the method. Since we did the enrichment step to capture only the pieces that come from active genes, we're only sequencing this part of the genome, which happens to be a relatively small part of the genome. So we don't do a lot of sequencing. And each sequence is an evidence for something happening. Does that answer the question? I'm checking how fast Yuval can type. Okay, thanks. Other questions?
We have a question from Nati.
Okay.
Nir, presumably you will talk about how you spoke about albumin, which has only a single origin, but what do you do when you get a mix of many different proteins that can come from—
Hold on to that thought, and we will talk about it in the future challenges. Yes, but you're right. You're 2 steps ahead of the talk. Okay. So if there are no other questions, I'll continue. So this was the introduction. It took 1/3 of the talk, so we are in good shape.
We actually have one more question.
Yeah, sure.
The student is asking, what's the reason that we want to separate between liver cells and other cells?
Okay, so liver cell was an example, but just to use this example to continue, if there's a person who suffers from something and we discover that there are many dying liver cells, then we might be able to pinpoint that the person suffers from some kind of medical problem involving the liver. Now, for this particular inference, there are much easier blood tests to do. But we're going to try to convince you that I can tell you whether that liver died from inflammation or from hypoxia or from other processes. And that's already information that's really hard to get from blood samples these days. And that's the direction we want to go after. But let's continue, and hopefully that will clarify itself. Okay? Okay, so in the rest of the talk, I try to put in four things, sorry. A bit about the technology, a bit about the biology of what can we see, some thoughts about how is this relevant to medicine and how we can use this. And since this is a CS colloquium, also try to open a discussion about interesting computational ideas that come from this kind of research. So let's proceed. Okay, so how are we measuring those pieces of chromatin? The challenge is that when we take blood and separate the cells and the liquid, which is called plasma, we're left with very little amount of cell-free DNA or cell-free nucleosomes. 1,000 genomes sounds like a lot, but actually it's nanograms, and there are many other entities there in several order of magnitudes more abundant. Some of them are proteins that tend to stick to things to make life more interesting. So actually, many people told us that what we're doing, they thought about it, but it's impossible to do. And so when somebody tells you it's impossible to do, it's a good sign that you might get something if you solve it. And here, I won't belabor the development, but basically what Ronen and later on the whole group working on it developed is essentially a reagent that is small beads with antibodies against the flag we want to capture, the green flag in this example. We put it in a tube with the plasma. This is an inert reagent, so it doesn't change the plasma. After a few hours, we essentially use a magnet to capture all those beads at the side of the tube, remove the plasma. We can then send it to other tests. And then we do a series of tests of processes on the bead that essentially lead to something we can sequence, although the amount of material is one that we cannot detect at this stage. We only later on will see if we actually cut something or not. So that's why it's a bit tricky. So there's a lot of many important details here, but the idea is essentially to use kind of the antibodies on the bead as kind of a hook to catch the fish that we're interested in, among all the other things that are in the plasma. And the immediate question that when one develops something like that is it working? Is it doing what it's supposed to do? And I can talk for half an hour about it, but I'll try to give a very short demonstration of one. I'm ignoring the chat because I cannot read long sentences while talking. I apologize. Is to look at kind of a picture that I'll explain what we're seeing in a second, but just to make the point, the top half of the picture, this area, is results that we got from blood, of plasma of patients, of healthy subjects and volunteers. And these are assays that were done with millions of cells from tissue samples from essentially volunteers or that contributed their body to science, or from blood collection from live volunteers by a consortium, worldwide consortium that did this kind of assay on cells, which is much easier. So what are we? So the point of the picture is that the two images look rather similar, but let's try to kind of work out what we're seeing here. So I'm going to hide everything but the red signal. The red signal is using one antibody that we now painted red. And those kind of peaks here are, if I remind you, the kind of marking of an area where there's a high peak like this. There were many fragments of DNA that we read that align to this part of the gene. And so an area that is empty, there is no fragment mapping to it, and a nice sharp peak is an area that we got dozens or hundreds of fragments mapping to that region. Now above, I put an annotation of some of the genes in this region. So for example, here is a very important gene called GAPDH. it's very short in this level of magnification, but we see that there is this mark at the beginning of the gene. Next to it is a gene called IFF01, which is going this direction. And next to the beginning of the gene, we see peak in all 4 samples, both the 2 from blood and the 2 from the repository. And the thing to kind of see here is first that the signal is very concentrated in those sharp peaks. This is how it should be for this mark. And that the peaks are the same peaks, essentially. Maybe there is a difference here. You see it here, but not here. But the peaks are essentially identical between the plasma with our protocol and with standard protocols on many cells. And in fact, an expert probably wouldn't have known that this is a different assay from different source if we hadn't told them. And the same can be said about the other marks. Some of them are more kind of spread out, so it's less easy to tell that it's similar. As this one is not that similar, it's kind of the noisiest, but for those 4 marks and more, we can capture the signals and map it to the genome. For the rest of the talk, I'm going to talk only about the red mark. And that's because of this property that the red mark is one that appears only at the beginning of a gene, not throughout the gene, but at a very specific location at the beginning of the gene. The antibody is considered like the gold standard in terms of quality. And because it shows up at the beginning of the gene, we can give that peak, this peak a name because we know it's the beginning of this gene and not other genes. So we can tie it to genes and then tie it to what is known about this gene from any other sources. So how do we work with this? So we're going to try to get away from these diagrams. We can scroll along the genome, but it will take us a long while to look at all the genome this way. And instead, we're going to, I kind of zoomed in on this region. We are going to define for each gene an area, which is marked in yellow. And all the fragments that fall on that area are going to be counted toward GAPDH. And all the fragments falling in this region are going to be counted for IFF01 and so on. Sometimes 2 genes are too close to each other and we're going to give it the name, it's one of the 2, but we're not sure. Which happens not infrequently, but not that much along the genome. And now so from this point on, I'm going to talk about numbers assigned to genes, and essentially it's the number assigned of fragments we found at the promoter of that gene, although I will not use this complicated sentence every time. Okay, so with that, let's look what we can do. So here is a visualization of jumping forward a year and a half. We collected a lot of samples. And what I'm showing here is a heat map that shows 14,500 genes across 268 samples. And each line is a gene, and the color yellow is, this gene has many more counts than in this sample compared to all the others, and blue, it's less than all the others in this thing. And this is just to show kind of a bird's eye view of the genome. And one of the points I want to emphasize, we're seeing real signal at those 15,000 genes out of the 20-some that I told you exist in the human genome. So let's kind of dig in a bit here. So the bottom half of this picture is kind of uniform. It's not precisely uniform, but it's kind of dull. And the top half is showing more interesting signal. And indeed, the bottom half is mainly housekeeping genes that tend to be uniformly used by many cells, while the top one is mostly genes that are specific to one or more cell types. So let's zoom in on that fragment. So we're zooming in on the 4,000 some genes that are showing interesting behavior. And without going who are the genes and what they are, because we're going to talk a bit about it later, I want to make 2 observations. One, there is a group of people that we call healthy. these are people who think they're healthy, and we believe them. But nobody is perfectly healthy, unfortunately. But I think they're all fairly healthy, or they don't know they have a problem. And we can see that they look more or less the same. There are a bit of changes along the columns, but basically we're looking at a fairly kind of uniform thing that is repeated. On the other hand, if we look at the other side of the matrix, we see here a whole bunch of diseases. Acute myocardial infarction at Kef Lev for Hebrew speakers. A collection of multiple different types of liver disease and a collection of cancer patients. And here we see, I mean, even without knowing what we're seeing, it's clear that there are many things going on. And so this already tells us that we can see a difference between a place where something is happening, a person where a bad process is happening versus a healthy person. But can we tell, what do we make sense of this? So we're going to try to break it down by several questions. So returning to Nati's question, what we are seeing in the blood, as I try to show in the animation, is essentially the aggregate of things that come from all the cells that die at a particular time. And in a healthy person, there's always death of all kinds of subtypes of white blood cells, of bone marrow cells that produce those cells. Turns out they are dividing a lot and dying a lot. A bit of liver, a bit of the cells that make up our capillaries, the blood capillaries, and few other cell types. And this signal we see all the time. And if we're going to look at pathological conditions, some pathological conditions are kind of like taking over everything, like some of the cancers we will see. But others are kind of, okay, they contribute some cells that are dying, but they are less than 1% of the cells of the contribution to what we are measuring. So can we detect something that is happening only in a small fraction of the cells that contribute to the big pool? This is relevant both in terms of understanding the limits of using this and also in terms of thinking about how to analyze this. So let's start with kind of thinking about this problem in terms of what are the relevant parameters. So if I want to detect a rare event, one of the things I need places where in that rare event are different than in other conditions. For example, we talked before about the albumin gene being different in liver cells from brain cells. So the albumin promoter with the mark that we are pulling is a location that is informative, is indicative about liver cell death. So one of the questions when we define an event is how many locations like that do we have along the genome? Is it just the albumin gene or it's 200 genes? So that's definitely a parameter, and as you might expect, the more places you have, the wider net you're using. The second parameter that is super relevant is the molecules coming from the cells with this event. What fraction are they of the total pool we're looking at? So if there are 100%, then we just need to take very few molecules to detect them. But if they're a very small percent, then we might need much more. So those are the two parameters we're going to look at, but there are other parameters that affect our ability to do this. And this is essentially the properties of the assay. I remind you, the assay is trying to capture these molecules because these are ones that are marked with the flag, and at the same location in all other cells, they are not marked in the flag. So we want to capture just these and not these. The problem is that assays, biochemical assays are never perfect. I called it enrichment because we are not capturing everything here. We're only capturing a certain subset. So there is a rate that tells us how good we are at capturing true things with the flag. And it turns out there is also a non-specific capture that is capturing things that are not our target, that happen to bind there weakly, and we happen to take them along. Now, this is, if the assay is good, then this thing should be much better than that. And we often can think of it as the signal-to-noise ratio. The problem that if we're looking at a rare event, then the pool of positive things is much smaller than the pool of negative things. So even if we capture very few of those in the sequencing, they might confuse us. So ideally, we capture most of the red molecules and very few of the ones that are not marked by red. And then we need to apply a test to decide, is what we're seeing really above the noise we expect based on the non-specific capture. I'm skipping a whole thing about how we estimate this thing. I'll just show you a simulation that uses parameters that are realistic for our assay in terms of the signal to noise and the capture efficiency and the amount of molecules in the blood. And what this simulation shows that as a function of how many positive, what fraction of the pool is coming from the cells I want to detect, how many locations do we need in order to be 95%, I think it was 95 here, 95% chance of detecting it. So it's kind of like, these are most likely we will detect it. And we can see, as you expect, that as the percentage goes down, this is a logarithmic scale, the number of locations you need is growing up. The two lines here are two different scenarios in terms of signal to noise. The point is that 1% we can capture with relatively few locations. Something that is 0.1%, we need more locations. But one of the things I want to show you that, for example, for liver, we have this many number of locations. So liver death, cell death, we can detect in less than 0.1%, based on this simulation. Since I'm kind of running slow time, I won't go into the detail. We did an interesting verification and showed that the simulation is relatively accurate. And then we use this test to see what we can capture or detect in healthy subjects. So in healthy subjects, we detect different types of white blood cells, which are the majority of cells that die, so it's not surprising. We see a very small signal from liver and T cells, but all of that signal is significant, and we believe, and so it's real. And we know that a healthy person has around 1% of the cells that die are liver cells, so this matches what we knew. This person, something happened, maybe he drank a lot the night before he came to donate the sample, we're not sure. I mean, but that's the kind of question we need to ask. But the other cells that are not supposed to die, indeed, we do not detect, brain and heart, for example. Now you can come and say, okay, maybe you simply don't know how to detect heart. You know how to detect liver, but your method of detection does not work on heart muscle cells. So we thought about it and we said, okay, we have a situation where we know that there is death of heart cells, of myocardial, and that's when a person is undergoing a heart attack, or the medical name is acute myocardial infarction. So we had several patients that were collected at admittance to the ER or immediately after the procedure, ChIP-seq, and we had all kind of varying levels of signals, but all of those were significant, while all of the healthy ones were not. Moreover, we can kind of look at the same person throughout time and see how after opening the area, a lot of DNA is released and the amount of signal is increased, and we can see all kind of other things, which I'll skip for now. So this is kind of a positive test case to show that we can detect damage to the heart if there is one. But can we look at the bigger cohort and see something interesting? And I'm going to look very briefly at cancer. And cancer is interesting because people are all worried about it, and it's also a situation where a lot of things are happening. And so we had the lack of collaborating with a very talented oncologist at Hadassah, Viad Zik, who after few test cases got really excited by our method, and essentially we raided the freezer of all the samples he could find and give us. So it's a lot of patients, some of them sampled multiple times, and they were not collected according to our standard, but most of them we managed to process, and they pass our quality control standards for using them. So we have 140 some, and you know, there's several dozens that are still waiting to be processed, but we were kind of cut short by the current crisis. So let's look at that. One of the immediate questions we can ask is, can we classify cancer? And here I'm showing you two ROC curve, receiver or operating curve, showing false positive versus true positive depending on the threshold, where the signal I'm going to use is either a signal for, these are all colon cancer, I'm sorry I didn't say that clearly. The signal, one of them is a signal like the signal we had for heart, we had the signal for digestive tract, because colon and rectum and intestine, they're all more or less similar in terms of the molecular signature. So we used the signature that we collected based on, again, this consortium, this large international consortium that look at different human tissues, define something that is indicative of digestive tract, and then applied it without any tuning to the cancer cohort and healthy cohort. And we got a very good classification. And this is mainly because healthy people don't have depth of digestive tract cells to the blood, only to the actual digestive tract, to the fecal matter. While people with cancer, the cells look like digestive tract cells, or like colon cells, and they die into the blood. A second thing we collected is from a measurement of RNA in many tumor tissues. And again, we use some tests to define genes that are high in colorectal cancer. And this signature did slightly worse, but also, again, without any training on our data. And this came from a totally different modality, not epigenetic data, but from gene expression data, for those who know what it is. It also performed rather well. So this is, for the optimist, this looks, oh, it's amazing. For others, is why do you have this crappy region here where you really can't tell the difference? And the reason we have it is because when somebody comes and say, I have a cancer patient and I collected blood, it's a moving target. There are certain time points where the treatment is actually working really well and the tumor is shrinking and almost disappears, and there's very little to find. In other situations, it's like a massive event, and it's kind of dominant. It's hard to miss. So just to get a sense of the complexity of this data, here is a kind of a timeline of one subject that went, they found a polyp long time before and started suffering something and came to the clinic with a very active cancer. I don't remember if it was in the liver or someplace else. She went into chemo with what is called a biological drug. The chemo worked and the cancer reduced, but apparently it had a side effect, so they switched the chemo, things got slightly worse. I don't know what exactly happened here because sadly we don't have measurements, but the clinical record, they go through a second line treatment, a third line treatment, and starting now, she's still alive and hopefully will continue to be so. They started a fourth line of treatment, and that's usually a sign that things were not going well. So there was a lot of tumor. And in the last few samples, we start seeing a reduction. So hopefully this treatment is actually working. The point is, if we collect her here, she's very close to the threshold we will use to put all the healthy subjects under. So it's very hard to detect cancer if we didn't tune our test. Yeah, we have a question. I'll answer it in a second. I'll just finish the the question. We can detect it if we train, but if based on the things that we not train in advance, this is harder to test, to capture, well, this is really easy. The question was why we don't expect higher signal when the cancer cells are dying, especially when the tumor is shrinking. So a tumor is actually counterintuitively, it's something that keeps kind of reinventing itself. The cell divides much faster than we would actually, the rate of growth of the tumor that we see in the body. So an intact tumor that is under control is constantly turning over, cells are dividing and dying all the time. When the tumor shrinks, there is a bit more of cell death because your kind of death rate is going higher, but it's not significantly higher than it was before. But now, since the mass is going down, you have a smaller mass to sample from. And when we look several months after inside the treatment, by this stage, maybe most of the cells that die are already out of the game. Okay, I hope it answers the question a bit. Immediately after you give the chemo, you see a massive death of cells, and it's actually something that is useful if I get to talk about, which I kind of doubt, but we'll see. Okay, we did all kind of things with this, and I think I want to skip it because I want to talk about some of the other things. We kind of try to take all the things that are cancer specific, build from it some kind of a low dimensional representation, using rather crude methods of groups of genes that are going up and down in most of the cancer together. We call them modules just to be fancy, and now we can look at each sample, say this is a name of a patient, idea of a patient, and the idea of the sample, and see the level of each group of genes in that patient. And this gives us all kind of benefits. For example, we can compare these two subjects that have more or less the same amount of cancer. And indeed, we see that this line and this line, which you can graphically see here, are more or less look similar. Indeed, all those are along the diagonal. But actually, if we look closely, these two modules of genes are higher in this subject than in this subject, and might indicate a subtype of cancer that is behaving differently in this subject. Another example, which returns to this patient we followed the before and after. So this is an early sample, and this is a late sample. Now the diagonal is much lower because the amount of contribution from cancer cells is lower. So it's like multiplying by a fraction. But certain things show up. So this is essentially a sign that we have less of this cancer. But this is a change in the phenotype of the subject. Something that was very low before suddenly is very high compared to everything else. In fact, these modules represent all kind of liver processes, and apparently the reason the chemo was changed from FOLFOX to FOLFIRI is because of the liver side effects. So this is what we see here. So this is indicative of a side effect that is, the cancer is down, but the side effect is going up. And these are two different subsets of functions of the liver so that we can make sense out of this. And again, I'm skipping a lot of details because I'm running out of time. Skipping all of that. I wanted to open up a discussion on data analysis. I showed you very few, essentially detection and this module business, but there are many things we can do with this data. And one of the questions, the first question is, what do we want to get out of it? Do we want to get out of it yes, no, sick or healthy? Do we want to say there's a tumor in the left lobe of the liver or something like that? Or want to say there's ongoing inflammation in colon tissue? Or we want to say this is ongoing inflammation that is responding to this kind of stimulus, and it's probably a sign that a bacteria from this phylum is activating the infection. This is a much more detailed description of what we are looking at. So the ability to do all of those things is limited by the signal we get. So if something is contributing a very small amount of material to the blood, it's really hard to reconstruct exactly what were those cells and how they look like, but we might be able to say something about them in terms of a yes/no question, like like the detection question we looked at the simulation. If we have more signal, we might be able to say more. And so this is constrained by the biology, but also constrained by our analysis method. And I try to divide things into two semi-orthogonal dimensions. One is how many samples we have of patients and healthy people and different variation in the population in our own assay. If we have few, to say that the heart subject has heart cell death, I had to use prior knowledge. If I didn't know what are the genes that are specific to myocardiocyte, the cells that make up the heart muscle, I wouldn't be able to do it. If I had many examples of people undergoing a heart attack or many examples of people with different types of cancer, I might, blindly, without any knowledge about the biology, be able to learn representations and classifiers that are telling me a lot. So as we increase the number of samples, we get ability to do all kinds of things, even though we don't understand the biology. But on the other hand, the more we know about the biology, the more we can do. So if we know a lot of biology, we can tell something based on very few genes and very minute signals in these genes. If we know more biology and have more samples, we might be able to do what Nati asked, which is kind of say exactly how many liver cells and how many heart cells and what was the status inside each one. And basically, if we are increasing both, we are getting into a higher and higher ability in terms of the interpretation. So in terms of the experimental lab, we're going this way. We're collaborating with clinicians, with Kupot Cholim, to try to recruit more people, run more assays, get more samples, be smarter. In this dimension, we are trying to use ongoing knowledge. So very briefly, just to give you a sense, there are three kind of types of data sets we can learn from. One is people using assays that are essentially measuring the same thing we are measuring, but on cells taken from the body. And those are like the consortium we used before, and there's a newer consortium that contains more groups in the world that is collecting more data also from different type of pathology, and that's directly relevant to us because it's the same modality. There is a much larger world of people who are measuring RNA, gene expression. There's thousands of samples out there in the wild. Here we're talking about millions. And in the last few years, people are measuring individual cells, and just this one site, you cannot see it because of the color. You can download 6 million cells from representing 200 studies that probably cover most of the human tissues, and they plan to essentially build an atlas of healthy humans, and there's atlas of different diseases. And that's important because this will be at the level of cells, not tissue. So if the tissue is composed of different types of cells, we will be able to know, is this the fibroblast in the liver or it's the hepatocytes in the liver, which are two different cell types that live in the same organ. The problem is this is a different modality, so we have to translate this very rich data into our media. And finally, there's a lot of other data that people are aggregating from literature, multiple experiments, and is super useful about what the gene encodes, when it's expressed, what is controlling it, and so on, that we would like to put into the analysis. So there's a lot of question on how to put this extra knowledge in a useful way into the analysis. And since I think I'm running out of time, although Yedid is very quiet, so I could keep on talking. I'll finish by very short discussion of medical applications.
We have 2 minutes on the clock, but we started a little late, so I guess that we have a few more minutes.
Okay, so I'll try to, I'm on the final trajectory. So the obvious thing people ask about, and for good reason, is looking at detection. You know, can we detect cancer way before there's any symptom that other people can detect? And this is kind of, there's one of the companies that does it, it's called Grail, because this is the holy grail of diagnosis. We believe we are very useful in that domain because we have all kind of properties I didn't get to talk about the amount of sequencing and the size of the signatures and so on. But when you want to go into that game, you have to collect large groups of subjects that some of them might develop a disease, and then see later on which one you detected in advance. So this is a much larger project than you can do at this stage of our development. One of our benefits is, as I mentioned before, cancer people use mostly mutation and other signatures of cancer, but we have ability to look at many other diseases that are totally don't involve mutation usually, like liver diseases, alcoholic liver syndrome or NASH is a set of diseases that many people in the Western diet will suffer about my age and onward. And it's going to be a bigger and bigger problem to detect it and then to treat it. Autoimmune disease is, again, we recognize them, but we might detect them earlier. And so we're collaborating with a whole bunch of groups. Some of them are listed here in terms of capturing diseases at early stage by looking at their earliest examples they have and then seeing if we can do something. Another idea, and this is an interesting idea, is that even if we don't have the perfect diagnosis, having a semi-informative diagnosis might be useful because we're doing a blood test. If once in a while, once a year, my blood is taken and sent to a test that doesn't cost much, and that test says, maybe you should do a colonoscopy, then my motivation to do a colonoscopy is much higher than the doctor saying, well, it's your time to do it because you didn't do it the last 5 years, so please sign up. It's much easier to do than colonoscopy, and it doesn't have to be 100% right to do it. Even if it increases the amount of subjects that we capture, we get a big win. In particular, since with the improvement of things like colonoscopy, the fraction of patients that are under the recommended age for screening are growing bigger because they're not screened. So somebody at the age 40 is not sent to do a colonoscopy as a screen, but it happens once in a while. And those people are now becoming a bigger fraction of the subject with the disease because they're not screened. But screening everybody under 40 with colonoscopy will kill the system. So having a test that is slightly more informative than what the doctor uses now might be useful for HMOs to make public decisions on who to send to the definitive assay, like colonoscopy, for measuring the device. There's all kind of issues of certification and approvals that are much easier for such screening tests. So it's also an interesting direction. Very quickly, disease monitoring. Many times we know somebody is sick, but we don't know what is happening inside. And so, we did a biopsy, we recognize the disease, we said to the subject, okay, to the patient now, if you keep this diet, your liver will function properly and everything will be okay. But in fact, once in a while, some of those patients, things go wrong, even if they adhere to the diet we gave. And we will know about it when that patient returns to us with already noticeable signals of liver damage, that show up in the blood test, in all kind of phenotypes of the patient. We want to capture it much earlier when the disease changes trajectory, but we won't do another biopsy on somebody who a liver biopsy is a surgical procedure, so it's not something you do kind of, let's do it. On the other hand, taking a blood test is a much easier way of following the disease. So if you can see the disease trajectory, whether it's getting better or worse, we can give feedback to the patient, keep the diet, it's doing really great, or wait a minute, we need to find out what's going on. Maybe we need to do more tests. Maybe we need to introduce other treatments. Similarly, for reoccurrence of disease that's like cancer that might reoccur after a while. But I think the most exciting way of looking at it is in terms of what we call personalized medicine, or I would say informed medicine. There are situations where we know that if a patient shows up molecular signature of certain things, we can treat that tumor by a drug. So there's something called HER2 amplification. It's a gene that is amplified in breast cancer, but also in many other cancers. And today, if somebody has a tissue biopsy that shows that, immediately they are approved to get this drug, and this drug works like a miracle on this kind of tumors, but it does nothing on other tumors. The problem is you might need to be lucky that the biopsy really caught the right location. And moreover, that as those indications become more, the growing number, that the doctor prescribed to you the test to, the pathology will test for HER2 amplification and not something else. I skipped the result, but we actually detect HER2 amplification and the effect of it in the subject from the blood. So this is something that the doctor can immediately say, wait a minute, here's an indication, I can make an informed decision. So that's a decision information before the drug, before the treatment. The second problem is, okay, I have to give a drug. You remember the time course of the patient. We looked at, there's like the first thing we give, and if that doesn't work, we move to the second line. There is kind of protocols like that. But the way to detect that something is working is to wait several rounds of chemotherapy and the drug dosage, and then do CT and see that the tumor shrunk. Okay, you will see that only 3 months later. But maybe the drug is not doing anything. The chemo is slowing things down a bit so that the patient is feeling better, but it's actually not getting rid of the tumor. So if we could have given a feedback immediately after the chemo, or 1 week after, the cells are dying because of chemo, but they're also dying because the drug you gave is really inhibiting this pathway that tells the cell to keep dividing, then this drug is working, keep on doing it. But if they're dying without responding to the drug, then this very expensive drug, the biological, the ones that do these are super expensive. You're giving this patient a drug that is A, not doing anything good, B, you're waiting a while before you do something else because you don't know that, and C, in terms of the healthcare, it's a really expensive drug, and there's all those fights on which drugs are, salad trufot and all those things that are because of the high cost of those drugs. So if you can give 1 dose of the drug and say it's working, let's go with it, or it's not working, let's stop. Similarly, side effects. Side effects are a huge problem in therapy. Most of the time you detect them way after a lot of damage is done. If you can detect it earlier, maybe you could have switched treatment before the patient suffers from kidney failure or from autoimmune response that kills the whole, the whole system, and it's really hard to detect, to essentially get out of it. So there are many things you can do by following the drug and talking about what we see about the drug from patients where it was useful versus patients where it was damaging or just not doing anything. And I think this is really where we're going to make a big difference. Okay, to summarize, I told you about an exciting method. I didn't tell you the story why we got to it, and you can ask if there's enough time later. it requires really small amount of blood. It's cheap on sequencing. It's highly sensitive, and it's super informative. And we essentially opened a fire hydrant of information. We're still learning how to drink from it, but we definitely see that it's a massive amount of water or information. And there are many questions we can do, including real application, but also basic science. There is a lot of things that people do not know because we don't know how to measure in a healthy human being. We are not going to open up somebody and do, even if they are volunteering, we cannot do to follow, for example, what time of the day there is birth of new cells and death of other cells and so on in different places in the body. Just an example. And so we're really excited about it. We're learning a lot of medicine as we go along. We're making a lot of collaboration with many good people. And there's a huge list of relevant people I mentioned. I'm listing, hopefully I didn't forget anybody important in this list. But there's also many, these are the people in the kind of the core publication that we're working on for the method, but there are many other people working on follow-up studies that we're working on. So thank you, and I run out of overtime a bit too much, I think, but and I see that my computer is about to die, so hopefully it will survive the question period.
We're a bit over time, but given that, you know, the room is not going anywhere, so obviously anyone who has to go can go, but if anyone has questions, then, you know, as long as Nir is willing to take these questions, we're going to keep this meeting open.
So in that case, I need to find power supply because my computer is out of battery in a second. Oh. But are there questions?
Okay, so far I can't see any hands being lifted. So thank you again.
It's my pleasure. Okay, so I won't connect to the computer. Anybody who's interested offline?
Sorry, I just said it's not clear how to clap hands here, but virtually we are.
Okay, thank you. But anybody who's interested, feel free to come to send me email. You cannot visit, but I'll be happy to elaborate offline also. Thank you.
Thank you.